So I left off with this question about serotonin. So serotonin is a monoamine. And the question is, how do monoamine inhibitors such as Prozac raise the concentration and duration of the neurotransmitter serotonin in the synaptic cleft? So MAO is monoamine oxidase inhibitors. So monoamine oxidase breaks down monoamines like serotonin. Monoamine oxidase inhibitors inhibit monoamine oxidase from breaking down monoamines. So if you can't break down the monoamines anymore, that means there will be more of them. So like I said previously, neurons interact with lots and lots of others, thousands, 100,000 other neurons. And they can do this, and it will lead to different types of interactions. So we'll go through each of these on their own. So the first is called summation. So summation is combining inputs to reach threshold. There are two different types. Temporal summation, there are multiple excitatory postsynaptic potentials at one synapse. Spatial summation, two or more different synapses are going to be simultaneously stimulated. Okay? So temporal, multiple excitatory postsynaptic potentials at one synapse versus spatial, there will be multiple at the same time. So either way, it's increasing the likelihood of reaching threshold and having an action potential. Presynaptic modulation is basically the addition of a third neuron right at that synapse. There's a third nerve cell. The third nerve cell can either inhibit or enhance neurotransmitter release. So with presynaptic inhibition, the third cell decreases or eliminates release of neurotransmitter. Presynaptic facilitation, it enhances the release of the neurotransmitter. So here you can see presynaptic inhibition. So here there's one neuron, here is the postsynaptic neuron, and then you can see this third neuron kind of butting in here. So what this third neuron is doing in inhibition is it is releasing its own neurotransmitter to this synaptic knob here, and it's releasing GABA, which is an inhibitory neurotransmitter. So it inhibits this presynaptic neuron here to make it less likely for this neuron to release its neurotransmitter. So here, this neuron gets inhibited and it doesn't release anything, so there's no signaling. And this is kind of the conclusion of the nerve section of our lecture. I'm going to do a quick central nervous system introduction, and then on next videos you'll see more details on central nervous system. So central nervous system is the brain and the spinal cord. If we remember, the nervous system itself is split into central nervous system and peripheral nervous system. Central being brain and spinal cord, peripheral being everything else. These divisions we'll go through when we talk about the PNS. This unit, we're just going to talk about the stuff in purple here. There's some definitions for you to know. So, ipsilateral, things are happening on the same side of the body. Contralateral, opposite sides of the body. Decussation is what happens when nerve fibers cross from one side of the brain to the other side. So for example, you guys have heard that like the left side of your brain controls the right side of your body and vice versa, and that's because the neurons decussate, they cross over. The commissure is where the actual decussation happens, so right where X meets the spot. So an example sentence here, fibers terminate on the contralateral side because they decussate at a commissure. My example that I use for contralateral is like putting on deodorant. So use your right hand to put it under your left armpit. Most people don't put on deodorant ipsilaterally, so they don't grab the deodorant in the right hand to put it under the right armpit. That's kind of a weird way to do that, right? A few other terms that I might use. Rostral means towards the forehead, and caudal means towards the cord. It's kind of like anterior and posterior, except in order to do comparative anatomy between species, like with a pig or something, we need to have rostral and caudal instead of anterior-posterior. There are lots of different sections to the brain. I don't care that you know the anatomy because we'll go through that in the lab, but you do need to kind of just have a general idea of where things are so that you know what each thing does. So brain is basically split into kind of 4 regions. There's the hindbrain, which is kind of the back of the brain here. There's the midbrain, which is the middle. And then there's the forebrain, which is like basically everything else. There's also the brain stem down here. In the forebrain, you can see there are 2 kind of hemispheres of the brain, 2 halves of the cerebrum. Anytime you see the word gyrus or gyrus, that's going to be the mountain peak. Anytime you see a sulcus, that's going to be like a little groove. Again, these terms you guys will need to know on the lab section, but not so much here. The cerebral cortex is basically the outermost part of your cerebrum, and then the nuclei are the deeper structures. You'll see here a cross section of the brain. Anything that's gray is going to be the cells, the neurons, and then anything that's white are those axons, the projections. They appear white because they are myelinated. And myelin is made out of mostly fat, which appears kind of white. Again, don't worry about the anatomy. These are just kind of FYI for you. The cerebrum is this big part here, cerebellum here, and then kind of the brainstem here. So with embryonic development, basically I just want you to know that it comes from the ectoderm. So the nervous system develops from the ectoderm. And already by the 4th week, before many people even know they're pregnant, the neural tube is already forming its 3 different regions, the forebrain, midbrain, and hindbrain. So just kind of know it's from the ectoderm and it happens already in the 4th week. This is kind of off topic, but folate is used in women who are thinking about conceiving. Folate's really important to make sure the neural tube closes. And the reason that women take this before they even know that they're pregnant is because by week 4, things are already changing in the development of the brain. So they need to be on this folate before they even know that they're pregnant. The brain is protected by 4 main features. First, the bone, obviously very thick. Second, the meninges. There are 3 different layers of meninges. There's dura mater, that's very, very thick. You see it here in pink. If we were doing the dissections in class, you would see This is literally like skin thick. It's very, very thick. Underneath that, arachnoid, which looks like a little spider web. This is here in blue. And then the pia mater is directly on the brain surface itself. Cerebrospinal fluid flows within these layers here to help protect the brain. And then the blood-brain barrier, I talked about a little bit previously when we talked about glial cells, but this helps protect the brain from anything that you're consuming or that's in your bloodstream. So this picture here on the right is a microscope picture of the meninges. So you can see here the dura mater is the outermost layer. There's a little space called the subdural space, and then there's a thin arachnoid layer full of blood vessels. Underneath the arachnoid layer is a subarachnoid space, and this is where cerebrospinal fluid is found. Underneath that is the pia mater that is directly attached to the brain surface. The pia mater provides blood supply to the brain itself. If you guys have heard of like subdural hemorrhages, or subarachnoid hemorrhages, these are like brain bleeds, and the more superficial they are, the less serious they are. The deeper they get, the more serious they are. So imagine if the pia mater is having a hemorrhage, that's going to be a big deal because that's supplying the blood directly to the brain surface, versus a subdural hemorrhage is kind of further away from the brain itself, so it wouldn't be as big of a deal. There's a disease you guys have probably heard of called meningitis. Anytime you see itis, that means inflammation, and meninges are the layers that protect the brain. This happens a lot in kids because of bacteria or virus, and I don't really care that you know all the specifics of this, but they have to stick a little needle into the back to get to that subarachnoid space to draw out some cerebrospinal fluid to see what the infection is caused by. The ventricles are little chambers within the brain that hold cerebrospinal fluid and kind of help to wash the brain. Oh, one thing about this, they're lined with those ependymal cells. If you guys remember, those are the ones that are secreting the cerebrospinal fluid. So this fluid is clear and it fills all the ventricles and the canals. It basically surrounds the brain to protect it. I just want you to know that where it's produced by those choroid plexus cells that are ependymal cells. The actual flow of cerebrospinal fluid we'll talk about in the lab, so don't worry too much about it here. Cerebrospinal fluid has 3 functions you need to know. Buoyancy, so basically it floats the brain. You don't want your brain resting on the skull, that would not be good. The bottom of the brain would lose its blood supply and it would cause damage to the brain. Second is protection. So literally it's fluid, so if you get hit in the head, there's fluid that the brain has to go through before it would hit the skull and cause bruising. Third, cerebrospinal fluid rinses away the waste. So it is being recycled throughout the day. As the previous slide said, brain produces and absorbs 500 milliliters per day. So this fluid is just rinsing away any type of waste. The blood-brain barrier is what I was talking about previously, and this is basically just the capillaries inside your brain being connected with tight junctions. So that means that these capillaries are not very leaky. Things have to be selectively transported. The only things that can be transported pretty easily: oxygen, carbon dioxide, alcohol, some hormones, and water.